Assalamu alaikum. Welcome to Wetlek. Today the topic of my discussion is fermentation in rumen. As the compound stomach of ruminants contains four compartments and one of these compartment is rumen. This rumen actually hosts different kinds of microbes like protozoa, fungi and bacteria. And due to the presence of these microbes, rumen is the site for fermentation. Ruminants consume green forages which contain cellulose and hemicellulose, and when these green forages are ingested and passed into the rumen, then they undergo microbial simplification within the rumen and also undergo physical digestion. So actually the bacteria that are present in the rumen actually possess enzymes, and these enzymes are needed to break the beta 1,4 linkage between the various sugars that make up cellulose and hemicellulose. So these human bacteria cause the breakdown of cellulose and hemicellulose which is contained in the ingested green forage. Cellulose and hemicellulose cannot be digested by mammalian enzymes, and it is the only task of bacteria which are present in the rumen. The cellulolytic bacteria that can break these bonds are actually the members of bacteroids Ruminococcus and Butyrivibrio genera, and these all are anaerobic bacteria. So when these anaerobic bacteria present within the rumen act on the ingested plants or green forages, then they break the beta 1,4 linkage of the plant cell wall structural carbohydrates, and these plant cell wall structural carbohydrates are actually cellulose and hemicellulose. So the bacteria break their beta 1,4 linkage and utilize the liberated hexoses and pentoses in the breakdown of cellulose and hemicellulose. The bacteria utilize the hexoses and pentoses which are being liberated by this breakdown reaction in order to provide them with energy. So these breakdown products are not passed to the host but instead they are utilized by the rumen bacteria. The anaerobic bacteria undergo the process of fermentation and produce volatile fatty acids that are then absorbed across the forestomach epithelium, and then ruminant uses these volatile fatty acids for energy. So the source of energy for the ruminants in this process of fermentation is actually volatile fatty acids. The pH of rumen is very important in regulating this fermentation process, because if pH is decreased, then at decreased pH bacteria cannot survive, ruminant bacteria cannot survive. The normal pH of rumen fluid varies with diet, so high forage diets promote higher rumen pH, around 6.5 to 7, and high grain diets decrease the pH of the rumen. At high grain diets there is increased rate of rumen fermentation, and as a result of this increased fermentation there is a increased production of volatile fatty acids, which result in decreased rumen pH. So high forage diets cause the pH of rumen to increase, on the other hand high grain diets cause the pH of rumen to decrease. So we have discussed the pH of rumen, and we have also discussed that the cellulolytic bacteria present in the rumen cause the breakdown of beta 1,4 linkage of the plant cell wall structural carbohydrates, and then utilize the liberated hexoses and pentoses to provide them with energy, and then they undergo anaerobic fermentation and produce volatile fatty acids that are used by the ruminant as a source of energy. Like cellulose and hemicellulose, the starches and simpler monosaccharides and disaccharides in the ruminant diet are utilized by the rumen bacteria, and they utilize it for their energy source. So very little amount of starch or sugar escapes the rumen for being absorbed in the small intestine. So the bacteria of amylolytic genera like Streptococcus and Ruminobacter digest this starch and sugar present in the ruminant diet, and cause the breakage of alpha 1,4 linkages in the starch, and then utilize the breakdown products of this reaction for its own source of energy. So these starches are utilized by the rumen bacteria as energy source. These bacteria then cause the fermentation of starches under strictly anaerobic conditions, So they cause the fermentation of starches and sugars and form lactic acid and also form volatile fatty acids. Sometimes cattle are switched from high forage diets to high grain diets, and when this abrupt change in the diet of the cattle occurs, then there develops a condition in cattle which is known as rumen acidosis. Actually, when cattle are switched from high forage diets to high grain diets on entering the feed yards, the amylolytic bacteria population within the rumen multiplies very quickly in response to the starch in the diet. So due to abundant amount of starch in the diet, the amylolytic bacterial population increases in order to increase the rate of fermentation within the rumen. So when the rate of fermentation is increased, then there is a production of large amounts of lactic acids along with the volatile fatty acids as a result of fermentation. Actually, the lactic acid has lesser pH than the volatile fatty acids. So this lesser pH of lactic acid makes it nearly 10 times stronger acid than the volatile fatty acids. So these increased rates of the fermentation within the rumen will produce high amounts of lactic acid and other volatile fatty acids in the rumen fluid, and this will cause the rumen pH to fall below the normal value of pH. When this human environment will become acidic, then the cellulolytic bacteria will begin to die and the rumen epithelium will be damaged by the accumulation of this acid. The bacteria which are dying because of this acidic environment will continue to release endotoxins, and these endotoxins will then enter the blood and will cause shock, and this whole condition that is produced is called rumen acidosis. So this condition of rumen acidosis occurs in cattle that have not been allowed time to adapt to high grain diet. So any abrupt change in the diet from high forage diet to high grain diet can cause this rumen acidosis. When the feeding regime of the animal is changed gradually, or the amount of the grain in the diet of the animal is increased slowly over a period of weeks, then it allows population of bacteria which are known as lactate utilizers to populate the rumen. These lactate utilizer bacteria belong to Celinomonas and Megasphaera genera. The function of these lactate utilizers is actually to metabolize the rumen lactate as energy source. So when grain diet is ingested and undergoes fermentation and produces lactic acid, then this produced lactic acid is metabolized by lactate utilizers as energy source. In this way, these bacteria prevent the rumen acidosis condition by preventing the accumulation of lactic acid within the rumen. The animals with very high population of lactate utilizers can be fed very high starch diets with little risk of rumen acidosis. As the monogastric animals or non-ruminants are hindgut fermenters, and they undergo the process of fermentation in their cecum and colon. So their cecum and colon contain same types of cellulolytic bacteria as are present in the ruminants, or in the rumen of the ruminants. So in hindgut fermenters, the cellulolytic material is fermented by cellulolytic bacteria in cecum and colon. But in these species, the starches and sugars in the diet are absorbed by the small intestine before they reach the colon. And in this way, the risk for very low pH in the hindgut fermenters is also reduced. A monogastric animal or hindgut fermenter obtains 4 kilocalories of metabolizable energy per gram of starch or glucose digested. On the other hand, ruminant obtains only 2.2 kilocalories of the metabolizable energy per gram of starch or glucose, and it is in the form of volatile fatty acids remaining from the anaerobic fermentation. In case of non-ruminants, the digestion of starches occurs in the small intestine. So in non-ruminants, starches are digested in the small intestine due to the presence of enzymes within the pancreatic juice and present in the small intestine. After this digestion, these starches and sugars are absorbed by the small intestine before they reach the colon. So there is no need of bacteria to digest these starches in case of non-ruminants or hindgut fermenters. But in case of ruminants, the amylolytic bacteria break the alpha 1,4 linkages of the starch, and then utilize the breakdown products for their own energy source. After this, they undergo process of fermentation, and they ferment the starches and sugars to lactic acid and volatile fatty acids, and these volatile fatty acids then are utilized by the host for energy purpose. Because of this reason, monogastric animals obtain about 4 kilocalories of metabolizable energy per gram of starch or glucose digested, and ruminants obtain 2.2 kilocalories of the metabolizable energy per gram of starch or glucose digested. So, in case of ruminants, the reason of this decreased extracted energy is the utilization of the starches by the rumen bacteria as energy source. But the monogastric animals derive very little energy from the cellulose and hemicellulose as compared with that of ruminants. So the ruminants can still obtain about 2.2 kilocalories of the metabolizable energy per gram of cellulose and hemicellulose in the diet. So in case of non-ruminants, cellulose don't supply the same amount of energy as the starches do. So the hindgut fermenters obtain about 4 kilocalories of the metabolizable energy per gram of starches by the digestion of starches in the small intestine, and they can also obtain about 2 kilocalories of the metabolizable energy per gram of the beta 1,4 linked plants structural carbohydrates, or they can obtain about 2 kilocalories of the energy per gram of the cellulolytic material after its microbial fermentation in cecum and colon. So the major source of energy for hindgut fermenters is actually the starches, which are digested in the small intestine, and the minor contribution to the energy in case of hindgut fermenters is contributed by the microbial fermentation of cellulolytic material in cecum and colon. Similarly, ruminants also obtain about 2.2 kilocalories of the metabolizable energy per gram of starch or glucose, and this energy is in the form of volatile fatty acids. And similarly, ruminants can also obtain about 2.2 kilocalories of the metabolizable energy per gram of cellulose and hemicellulose in the diet, and this energy is also in the form of volatile fatty acids. The bacteria present in the rumen are also the source of proteins for the host animal. So the rumen bacteria have the ability to combine with nitrogen, to combine nitrogen from ammonia or urea with carbon skeletons that are liberated from the dietary carbohydrates, and in this way they form the amino acids that make up the protoplasm of animal. Similarly, when the bacteria dies or it moves into the small intestine with other digesta, then the proteins within the bacteria can be digested by the mammalian proteolytic enzymes, and amino acids are used by the cow. So the digestion of the bacterial proteins by the mammalian proteolytic enzymes releases amino acids, and these amino acids are then used by the ruminant or host animal. So the proteins that are produced as a result of that are produced by these rumen bacteria are considered are considered of very high quality, and their amino acid profile is almost identical to that of the muscle and milk. So it helps the conversion of these amino acids into the milk and meat by the host animal. There is also a disadvantage of the ruminant for the ruminants, and that disadvantage is that much of the protein that is fed to the ruminant or the host animal is utilized by the rumen bacteria. Actually, the rumen bacteria find it energetically more efficient to use the preformed amino acids rather than making the new amino acids for the host animal. So the rumen bacteria prefer to use the preformed amino acids that come along with the dietary protein. The dietary protein that the rumen bacteria can break down in order to obtain amino acids is referred as the rumen degradable protein. So in the ruminants, if the protein is rumen degradable, then the essential amino acids in the proteins are utilized by the microbes or these rumen bacteria, and in this way these essential amino acids are not provided to the host animal. These lost essential amino acids can only be provided to the host animal if they are recovered in the form of microbial protein that enters the small intestine. So the essential amino acids contained in the dietary proteins can only be recovered by the host animal if the bacteria enters the small intestine, and it is digested by the mammalian proteolytic enzymes, and then these essential amino acids are released and they can be used by the host bacteria. So this is the only way to recover these lost essential amino acids. In monogastric animals, this is not the case because they contain microbial populations only in the cecum and colon, and most of the absorption of the digested matter occurs in the small intestine. So the amino acids in the proteins which are supplied to the monogastric animal are not lost because they don't contain bacteria in the small intestine. So in this way, these amino acids are not lost in case of monogastric animals by bacteria. So in monogastric animals, it is critical to feed high quality proteins in order to supply the animal with essential amino acids, because there is no worry of the loss of amino acids by the consumption of these amino acids by the bacteria. So the monogastric animals should be fed high quality proteins in order to supply them with essential amino acids. In case of ruminants, not all of the dietary proteins fed to the ruminant are degraded by the rumen bacteria for their use. So the dietary ingredients of the proteins vary in different proteins, and the degradability of the protein by the rumen bacteria depends upon its dietary ingredients. So all the dietary proteins fed to the ruminant are not degraded by the rumen bacteria for their use. So most of the proteins which are found in the typical feedstuff contain between 25% to 80% of the rumen degradable protein. So these 25% to 80% of the rumen degradable protein which is present in the typical feedstuff is degraded by the rumen bacteria. And on the other hand, some dietary proteins are not degraded by the rumen bacteria, and those proteins that bypass the rumen bacteria and that are not digested or not degraded by the rumen bacteria are known as rumen undegradable proteins. So these rumen undegradable proteins are not degraded by the rumen bacteria. They can only be digested in the small intestine, and their products are then absorbed in the small intestine. So if this human undegradable protein is of high quality, then it can prove to be an excellent source of essential amino acids for the host ruminant. The proteins that are supplied to the hindgut fermenters are also digested and absorbed in the small intestine, in their small intestines. Similarly, amino acid transporters are also present in the colonic mucosal epithelium in case of horse, and it means that some amino acids are also absorbed or transported across the colonic mucosal epithelium. And because of these amino acid transporters, presence of amino acid transporters in the colonic mucosal epithelium, it means that the microbial population of the colon of hindgut fermenters also contributes to the production of amino acids.